Okay, good evening, everyone. Uh, we are going to be talking about human genetics. Uh, it's actually a branch of biomedical science that studies the structure, the function, transmission, variation, and expression of genes in humans, and how this influence health, of course, and of course the diseases. Um, if you look at it, it actually connects molecular biology, cell biology, biochemistry, embryology, pathology, and even clinical medicine together. Okay, so clinically, human genetics explain why diseases run in families. Somebody inherits something from someone, so why some congenital anomalies occur, why individual responds differently to drugs, and why certain diseases present early, why others even manifest actually later in life or even in several generations ahead. So it's actually a major milestone in a major milestone in human genetics, or what I would say a major milestone in modern genetics was the Human Genome Project, which successfully mapped the complete sequence of human uh, DNA. And this project actually laid the foundation for precision medicine. Okay, so now there are just like every other discipline, there are actually basic terminologies in genetics. Okay, for example, we have a gene. From ordinary biology, you know that a gene is the functional unit of hereditary and consists of actually specific segments of DNA that codes for a protein or functional RNA molecule. When I mean RNA, I mean ribonucleic acid molecule. So mutations in genes may actually lead to diseases. All right. Now the genome, on the other hand, refers to uh, the entire material of human of an organism, and the human genome actually contains, if I can remember my number correctly, approximately, so I'll use the word approximately, 3 billion base pairs of DNA. So that's that. And then of course there's something called a chromosome, which is a highly organized structure that is composed of the DNA and proteins. And when I say protein, I mean histones that actually carries the gene, the one I mentioned. And from what we've known, even from ordinary biology, humans normally have 46 chromosomes arranged in 23 pairs. You inherit from your both parents. And there's something called an allele. Allele, if I can spell it correctly, I think is ALLELE, which is an alternate form of gene. It's actually an alternate form of gene that is located at specific locus of a chromosome. It means different alleles may actually produce different variation in traits, which means someone can actually, if a father is fair, all right, the first son can actually be a little bit fairer than the second son. All right. Now, there's also something called the genotype, which is the genetic constitution of an individual, while the phenotype refer to the observable presentation. Now, importantly, we can say that um, genotype does not always directly predict phenotype, and this is actually due to environmental influences and some modification. I think epigenetic modification. Of course, environment actually influence how someone inherit some traits. Okay, there's something important that viruses do, another agent. Since I mentioned environment, there's something called mutation. A mutation is a permanent alteration in the DNA sequence. It may be pathogenic, might be benign, even sometimes it might even be protective. Okay, there's another one called polymorphism. Polymorphism is polymorphism is a common genetic variation in a population, and of course it's often distributed to different places in drug response or even disease susceptibility. Sometimes you see someone they're able to take like 10 bottles, without getting drunk. Meanwhile, someone else can't even take one. All right. Now, let's talk about organization of the human genome. Remember, I said that the human genomic or genome project is actually a very important project. In fact, one of the modern revolution. Okay, so now talking about the human genome, it contains approximately 20,000 to 25,000 protein coding genes, and these genes are actually distributed across about 23 pairs of chromosome, including 22 pairs of normal chromosome or what we call autosomes, and one pair of sex chromosome. Sex chromosome meaning XX in female and XY in male. Now, at the molecular level, genetic information actually flow from from uh, from DNA to RNA to protein. This concept is known as the central dogma of molecular biology. All right, so DNA stores genetic information, then move to RNA. RNA stores no DNA stores genetic information. RNA transcribe this information more like a computer, and then the protein executes cellular function. So at the chromosomal level, the DNA is packaged into what we call the chromosome, which actually become visible during cell division. If you remember in biology, in ordinary biology, during mitosis and meiosis, you see that at a particular period, the chromosome become very visible. Okay, so structural or what we call numerical abnormalities at this level may actually result in syndrome. Whenever there's a problem, for example, whenever there is a chromosome is short in one, or whenever there is a damage in chromosome, it can actually result in a lot of syndrome. Now, at the genomic level, the entire DNA sequence, both including coding and non-coding regions, so this is actually very important, is actually considered. And now only about 1 to 2% of the genome codes for the protein. The remaining majority actually consists of the regulatory sequences, introns, and non-coding RNAs that control gene expression. Now, let's talk about type of genetic variation and diseases, because this is actually where it gets interesting, and you can even say important. Now, genetic disorders can actually be classified into two or three major categories. We have the first one called the monogenic disorders. This one actually results from mutations in a single gene, and typically, it follows the what we call the Mendelian inheritance patterns. Okay, example is a sickle cell disease, for example. Is sickle cell disease. We have if I can remember some of these diseases, cystic fibrosis, even Huntington disease. It happens like that. It follows the Mendelian inheritance pattern. So in this condition, a defect in one gene is enough to cause a disease. Once there's a defect in one gene, is enough to be catastrophic. Okay, then there's another one. The second one is chromosomal disorders. This one arises from abnormalities in chromosome number. A chromosome has to be short, okay, or structure. So an example of this is Down syndrome, for example. And Down syndrome actually results from an extra copy of chromosome of 21. That's chromosome 21. And clinically, this condition is actually associated with intellectual disability, although there are ranges. All right, some intellectual disabilities are worse than others. All right, we also have hypotonia, of course, characteristics, facial features whereby there will be tiny or slanty eyes, and congenital heart defect, of course. Now, there's a third one called the multifactorial. I'm sure this one is very popular. Even chromosomal abnormality is actually very popular, but this one is another popular one called the multifactorial polygenic disorder, which of course results from interaction between multiple genes and environmental factors. All right. Now, uh, 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 you know, we have hypertension, for example, type 2 diabetes mellitus, uh, cleft palate, or cleft lip and palate, and even many cancers, can actually follow this multifactorial disorder. Okay, now what are the patterns of inheritance? Patterns of inheritance.